I, Dr. Atri Shahana, welcome you all to this international webinar organized by Department of Zoology in collaboration with IQSC, Michael Mudhusudan Memorial College, Durgapur. Now, at the very beginning, I want to welcome the person without whose constant support this webinar would not have been possible. Our Honorable Principal Sir, Dr. G.M. Helaluddin. Now, I welcome Sir. Welcome you, Sir. I request our Principal Sir to please share a few words with us. Over to you, Sir.
Okay. Just a minute. So, good afternoon, everybody. Present in this international webinar organized by Department of Zoology of our college. Now, warm welcome to all our students, faculties and others, and also our resource person, Dr. Bodhaditya Chaudhary. Welcome, sir.
Thank you.
This is our second webinar by this department. First one was national and this one is international webinar. So I think our students are very much excited in joining this session. Because the topic selected for this session is very much important. I think this is to some extent related to behavioral neuroscience, and I don't want to elaborate this because this is not my subject, but the topic I have seen, that is the search for King Solomon's ring in the new age of behavioral neuroscience. So I think I strongly feel that our students, participants will be benefited to a large extent with their active participation and interaction during the presentation. One thing to be remembered, that every academic activity is important for their development. That may be seminar participation, maybe paper presentation, article writing, field survey, or many more such things. So they can develop them in this way. I hope all of our participants will be enriched through this valuable lecture session. So they will be careful, sincere in attending this session. I think they will actively participate. Means interaction session is most important, not only listening. The interaction is most important. Now, special thanks to once again Dr. Bodhaditya Chaudhary for sharing his valuable time and expertise with us, for our students, faculties. And lastly, I hope the grand success of the event, the effort of our department, the faculties, Dr. Sahana and his other faculties of the department and staff. So thank you very much. And I think next I over to Dr. Sahana.
Thank you, sir. Thank you very much for your joining and you inspired us a lot. Thank you very much, sir. Okay,
Thank you.
now we are coming to the main course of our webinar of this webinar, and we all know the topic is the search for King Solomon's ring in the new age of behavioral neuroscience. Communication is very crucial for any living being, maybe for a human being with another human, animal with another animal, or between human and animal. From the very prehistoric era, human evolved his communication skill in great extent, not only within themselves, but with other non-human species too. One such great example is King Solomon, who was a Jewish king of before Christ era and was renowned for his extraordinary capabilities like speaking with animals. He had a ring, and it was thought that this ring rendered the king such power. With this mythical background, the title of this webinar deals with behavioral neuroscience, which we all know that it covers neural and biological basis of behavior. Therefore, I guess we are going to experience a great journey into the field of animal behavior, linking it to neuroscience. We are fortunate enough to have with us Dr. Budhaditya Chaudhuri to enlighten the topic. Welcome, Dr. Chaudhuri.
Thanks.
Dr. Chaudhury's research interests encompass genetic and neurobiological understanding of animal behavior. He obtained his master's degree in zoology from Vishwabharati University, India. An Indian Academy of Science Fellowship funded his thesis on metapopulation dynamic modeling in Drosophila in the lab of Dr. Amitabh Joshi at the prestigious JNCASR, Bangalore. He carried out his PhD work at the J.P. Scott Center for Neuroscience, Mind and Behavior at Bowling Green State University, where he established quantitative behavioral methods to study large-scale open-field movement in dogs using GPS data loggers and computational analysis tools. He joined at the neurobiology department at Harvard Medical School to study molecular basis of social behavior in Drosophila in 2012. During this time, he identified critical developmental windows and transmembrane transporters that modulate social behavior in Drosophila. While at Baylor College of Medicine, he developed a high throughput behavioral pipeline to study and quantify long term behaviors using machine learning platforms. He joined Advanced Science Research Center, City University of New York, to study sleep regulation in Drosophila. He is working on building video-based real-time behavioral platforms to dissect the relationship between the homeostatic and the circadian control of sleep time. Outside the lab, he can be found in volleyball and tennis courts, independent bookstores, and art museums. Last but not the least, I want to share with you that Dr. Chaudhary is a very good friend of mine from my graduation days at Vishwabharati. So it's my pleasure to have him. Now I invite Dr. Chaudhary to please deliver his speech. Over to you.
Yeah, thanks, Atri. Thanks, Dr. Helaluddin, for a wonderful introduction. And yes, Atri said we go quite a few years back. I did my master's from Vishwavarati, where Atri was a student as well. So this is really nice to reconnect and have the opportunity to talk about the work that I have been doing in the last few years. Let me share the screen, and then uh, we can start.
Okay, okay.
Uh,
Yes, yes. Please, yes.
with, yeah, and then I'll talk more as we go through. Let's see if you can see it. I don't know. Can you guys see it? Um,
Hello, hello. Okay.
Hello. Yeah. Can you guys see it?
Hello, hello. Yeah, yeah,
Yeah.
yeah. Yeah. It is seen, it is seen. Yeah.
Okay. Okay.
Clearly
Okay.
it is seen. Yeah.
I can see my screen. So yeah, like Atri said, the topic is on behavioral neuroscience, and Atri gave like a really nice introduction on King Solomon and then the mythical ring. And the idea in neuroscience have been for a long time to be able to communicate. Communication is important, and some of the figureheads of this approach have been Konrad Lorenz here on the left, who discovered imprinting, and then he would imprint these gray lag geese to follow him around. And he found this very short developmental window, I think around 16 hours right after birth, where the geese get imprinted on following anything that moves around them. And so he would imprint the geese and then they would follow him around. But since those times, neuroscience has come quite the distance. And what you see here on the right, this is a very recent work from Janelia here. So this is called the fly hemibrain. So flies have about 100,000 neurons. So Drosophila, fruit flies, about 100,000 neurons. And now people in collaboration with Google and other high-tech software companies have been mapping these neurons, right? And so what you see here in the movie is called the hemibrain. So this is a fly's higher order processing center, and this cluster, which looks like kind of a mess, this is about 25,000 neurons. Every single neuron, every single connection, they have mapped. And what's coming here in between these neurons and the structure are those higher order processing centers and how the neurons wrap around these centers in the fly brain. So we know now with very good accuracy and precision, every single neuron, their identities, which other neurons they connect to, and how they carry out higher order processing. But what is in the middle here gets little attention, and then it's starting to get there. I feel like last few years have been really good for behavior quantification, and kind of making the bridge between these naturalistic behaviors and how the brain and the body carries out um, these behaviors by molecular pathways, by neural pathways, by genetics, by mutations. So my talk will mostly feature this middle section where we go through the different techniques people are using these days to study behavior, and how that um, starts to give you like the more fuller picture of the understanding of behavior. Okay, so I generally try to put my acknowledgement slides in the beginning while everybody's still awake. So I have been very fortunate to work with people who are really, really great mentors. And I think this often gets underappreciated, how important having a good mentor is in developing your own research career. So I first did my PhD with Robert. Here, Robert used to be a programmer for Apple using his skills in the early 70s. So he had a really good computational background, and my interest in computation and animal behavior started with him. Ed is very old. Ed is 85 now, but he still comes into work every day. He works actively, and I think that's the only thing that keeps him going is doing science. Ed is really, really nice, and I learned basically Drosophila behavior from him, and he established the Drosophila aggression field, or re-established in the early 2000s. There have been 3 or 4 papers in Drosophila aggression before, but after his papers, the field really exploded. And Ori, I currently work with Ori, and who is also a great mentor. He's at ASRC in the CUNY, and he is really supportive. It's not that easy right now these days with the pandemic. because I feel like this often doesn't get appreciated enough, is lives are difficult during a pandemic, and it's very important to work with somebody who not only is a great scientist but also a great person. And I think Ori is all of that. So these three people are definitely important in my scientific trajectory right now. And so to get into the talk, let me give you a brief outline of what I'm going to talk about. So I will talk about like a very, very brief history of animal behavior. It's kind of like the journey into uh, into this field, but I'm not going to get much detailed into it because I know in India, especially in zoology departments like yours, you have very capable faculty who are very good at giving you the historical perspective, how things came about, all the conditioning experiments. So we have in India in zoology departments very, very strong background in behavioral teaching. So I'll just give a very brief tour and not belabor the point. But I will start really talking into the new tools of the trade, which are how behavior is being studied these days. So there's this idea that if you could open up like animals, there would be these wires coming out. And if you understood every single wire, you would understand behavior, right? And there's this TV here. There are these USB ports, plugs, phones. So it's very in the cross sections of technology, and even though that's important, like I mentioned in the first slide of those neurons that are mapped, every single one of them, you still need to understand context. You still need to understand development, understand how behavior is studied. So we will talk about all of those a little bit. Uh, I do want to mention machine learning because I feel like it gets a lot of attention these days. Deep learning, machine learning, computation, and I want to demystify it just a little bit because I feel like when people talk about machine learning, there used to be this idea, actually during Lawrence and the early times, that if you give stimulus into a black box, you'll get a response out of a black box, and that black box was behavior with fixed action patterns. But I think it's more than that, right? I mean, so here in this picture of all these dogs, you have different dogs, but if you look carefully, there are patterns, right? So this one and this dog, they are similar. The stretching dog is kind of similar to this one, but just the other way around. And so machines are basically computational tools, and they look for patterns, right? And they're very good at identifying patterns. And in the end, that's all they're doing. They're identifying patterns, they're matching patterns, and they're giving you an output based on those patterns. And those outputs are only as good as the inputs you're feeding them, because if you feed in junk, you will get a junk result. So I think it's important to understand a little bit of machine learning, at least the basics of it. And then using some computational tools, I will show a brief study of behavior as relics of time, because uh, behavior is not just happening only when somebody is looking at it or when one is quantifying it, right? I mean, behavior has these developmental aspects, that this evolutionary aspect to it, and I will focus on these. Focus on hunting dogs, and there was this idea that dogs are um, selected from wolves, and they have been selected many times over through artificial selection to do. You have different breeds now, like hundreds of breeds, who have been selected to either do field work or just look pretty and stuff. But if you focus on particular behaviors that these dogs elicit, I think there are patterns that emerge, that may be relics of the past, maybe a history of what came before. And I will end with what I mostly work with is fly aggression. So what you see here in the movie is are two flies. These are two male flies because female flies don't fight in conventional patterns. But these males, if you give them limited resources, like here, limited food sources, and one of them is exposed to the other one for the first time, they will start fighting. And what they are doing here, you see these very fast snapping behaviors, those are called lunges. And most of fly aggression looks at those lunges because those are uh, critical at establishing dominance, at establishing who is going to be the winner, post-win effects, and all of that. And I'll have a section in the last bits of my talk where I can show that this is a very complex set of behavior, and using machine learning, how you can try to automate it. Because I mean, if you look at here, so there's going to be lunge from this guy. So write that 1, 2, 3, 4, 5, 6. So I mean, I can see it because I've looked at it many, many times, and I can recognize this pattern. But how a machine does it, who has no idea about these videos, right? So we will talk a little bit about that. And so that will be the last parts of the talk. And I'll talk about both modulation of behavior and persistence of behavior. So there are two subsections of that. And with that, I will end the talk. So to give a very, very brief history, and if there is one thing that you should take from this talk, that the nature versus nurture debate is dead, right? I mean, there are a lot of articles that have been written in the past about nature versus nurture. There is no nature versus nurture. It's always nature and nurture. You cannot have one without the other. And to understand why people talk about nature versus nurture, we have to look back a little bit in history and geopolitical history, is look at so the idea of nurture, that you can learn anything, it's mostly an American idea, where you have these. So this is like a mouse in Skinner's box, where the idea is if you press a lever right here, like a food pellet will drop, and then by trial and error, a mouse figures out how to press this pellet, and then it keeps pressing this pellet and to get the food, right? Here it is a Thorndike box, which is a puzzle box. And when you put an animal in there, a cat, the first time it tries many different things to open the box, and it opens the box and it gets out. But if you put the cat in the same box over and over again, you realize that the time it takes to open the box slowly decreases. So it's by trial and error, it figures out how to open the box. So that was the idea of learning over repeated trials. And in the end here, I have these, it's called the Little Albert experiment, where generally, let me play this movie. Generally, babies like fluffy things like rabbits as such. And so this experiment was, what I'm not showing you before is the experimenter, this guy. So these are very cruel experiments, by the way. He had a big like gong, and every time the rabbit was exposed to the baby, he would hit that gong, and with a really loud noise. And over time, this is fear conditioning. Over time, the baby learns to fear this rabbit. So every time you put the rabbit on the baby, it starts crying. And these are all the early psychoanalysis studies. And so these are very American approaches, which says, the important thing is nurture. There is no genetics, but you can be anything you want to be if you just learn. On the other end of the spectrum are these European scientists. So this is Konrad Lorenz with his gray lag geese following him that he imprinted. His idea being, you can imprint an animal, and so this is all patterns that they do, and this is inherent, and doesn't matter. He can let like geese follow him even though he's not their mother. uh, Nico Tinbergen was another famous scientist who did fixed action patterns, and he showed that these patterns, when you start these patterns go all the way, so they fulfill. It's very hard to stop the pattern midway. And uh, and he studied these pecking behaviors, these the carrying the eggs in the nest behaviors. And then you have Carl von Frisch, who studied these honeybee colonies, literally taking measurements and time to figure out this waggle dance, where it's a very, very complex dance that honeybees perform in order to communicate where food sources are. And these nature ideas that, okay, animals are born with certain capacities and you only are what you are born with, is a very European idea. And mostly the Second World War was fought on these ideas, that on one side you have these people who are very strong proponents of eugenics, saying, there are higher, the idea being there are higher races and lower races, also kind of stems from this idea of you are what you're born with, right? And on the other end, you had these transatlantic people who are like, oh, no, you can learn. And so the truth is somewhere in the middle. A lot of the times people don't mention that Tinbergen also did a lot of learning and memory experiments, which are very classical nurture experiments. And also people on in the US who are very nurture oriented people did strong genetic experiments which crossed borders. So when you study historical ideas of nature and nurture, keep those ideas in mind as well. The geopolitical idea, why this still gets so much press. And so the new tools of the trade of studying animal behavior are these video cameras. And I think Shobujda before me talked about these GPS receivers, which are these tiny things that you can now put on animals as the animals move. This is kind of an input of the logging of behavior. And then you have this behavior processing part where you can use MATLAB or Python based codes to analyze those data that you gather from these movements. More and more these days, you have these. So this is a Arduino and this is a TLC board. So what Arduino does is, this is basically a very small, like a computer, and these chipsets and analyze behavior in real time. And these TLC boards, they can relay those behaviors into stimuluses and outputs. So what you see here, so this is basically, you have an Arduino, you have a TLC, maybe this one has a Raspberry Pi as well. And all of these wires are connected to these tiny motors. And based on what behavioral input that you're getting, you can write codes and train these machines to carry out certain behaviors, right? And so it goes from input to output to real-time feedback loops using computational and machine tools. And so this is just to give you an example of what people are doing these days. So these are all animals that are being tracked. So these are video-based trackings. In the left, you have these mice in a box. Here you have the whole animal tracking. And so you see each mice have a directionality. It knows which is the front of the mice, which is the rear end of the mice. And you would see there are tiny dots along the axis of the animal's body. And from each dot, each second, tons of data are being collected, right? So now you have lots and lots and lots of data. And remember, when you see these, the mice in a in a cage being monitored and tracked, these computers don't know what is a mouse. They don't have any concept of a mouse. Yet it can maintain identities of each mice. This kind of yellow tag tracked mice crosses over to this magenta, but it maintains its identity. So it's smart in the sense that it doesn't lose identities, and it gives you a lot of data. Here you have tiny capillaries of this animal being tracked, and you can, so I think these experiment, they had certain stimulus here, odorants, or it's getting fed something, and then basically noticing how the animal moves and how the animal interacts by this vibrissae. You have very typical paw drawing a liver. So imagine kind of like a Skinner's box experiment, and you can see how each finger of this animal is being tracked. And you can, the things you can get from those type of experiments is how much the animal grabs around the liver. And then you can study with different mutants where those grabbing behaviors are impaired. You have a very classic a wheel where the mouse is running, and then you can study gait, you can study posture, and finally you have these Drosophila where now you're tracking all body of this Drosophila from multiple points, and each of these points are giving you information, right? So for instance, this is a female Drosophila who's laying eggs, and you can study grooming behavior, like in the ovipositor, it grooms, and all of these different things. So these are, I would call them the first order tracking experiments where you can get all of these data. And so you can get a very, very rich data set. And this is through by using Deep Lab Cut. This is like a software. This is like a freeware. Anybody can download it and use it. Another paper that came out literally last week goes one step farther. It uses the same ideas of cameras, but now it tracks animals in 3 dimensions. So, right? I mean, you don't have to focus much on all of these schematics, but basically the idea what's happening is there's a camera, there's a central animal, and based on the position of the animal using 3 cameras, the close-up camera can change. And so you have this servo control. So servo is just a motor that controls this camera. Based on all of this information it's receiving from 3 other cameras, and keep the animal in focus and basically move around. And what this close-up camera does is gets these images where you can then reconstruct the posture and track the animal in 3 dimensions. Like here, they have tracked 2 animals in 3D, which is one is red, which is one is blue. And so these are very, very highly processing. You need high processors to do these experiments, especially if you're doing it real time. And their paper talks about how you can do this real time. And when I say real time, that means as you're getting information in on what the animal is doing, you can give the animal feedback within milliseconds. So that requires quite high processing power. That's why it has these 3 GPUs, right? So this is quite an intensive experiment. But you can also do, people are also doing it these days using cloud computing, where you basically have something like Amazon cloud systems, Amazon cloud services, and you can just tag it to the cloud, and you can process your data there. So all of these approaches in behavior, they are opening up. And they will be more and more in coming years, because people are really now starting to focus on animal behavior. Okay, and so the idea is, what do machines learn? I mean, we have this idea of, you know, if a machine is just a bunch of these knobs and levers, but when it really boils down to it, there are very specific things these machines are learning, right? I mean, and you can subdivide it in 3 major classes, which is unsupervised learning, which means the machines learn without any input from the observer or the experimenter. You have clustering and dimensional reduction, which are just statistical terms to tell you that when you start with a lot of data, how you can reduce the number of data without actually reducing the quality of your experiment. You have supervised learning, where as the computer is learning these behaviors, you have a supervisor, which is a behavior which feeds back into the machine saying, okay, you did this, which is right, and you did this, which is wrong. And as the computer just goes through many iterations and loops of what is right, what is wrong, it gets a probabilistic idea of getting or doing the right calculations over time. And the third major one is reinforcement learning, which I showed you before in these feedback loops. People use robotics cameras to learn from the environment and then just feedback using loops. So these are the basic tenets of machine learning, and I'll give you very brief very brief examples of all three of these, and basically just to tell you that it's not rocket science, right? I mean, people like me are doing it, and so if people like me can do it, people like you can do it, and there is no sort of this high praise or this almost devout attention to machine learning. It's not really that. It's fairly simple. And so the first story, as Atri mentioned, I did my PhD on dog behaviors. I will talk about that and talk a little bit on computation. So the idea of dog behavior is very anthropocentric, and by anthropocentric I mean people always try to kind of make them relatable, right? More than what the dog is thinking, they would focus on what I am thinking, what the dog is thinking, right? So it's very focused on the human experience. But if you look at history and what fossil records, not fossil records, but all these historical records show us that, and now genetic analysis, that these gray wolves, they cohabited with humans for at least 100,000 years ago, and then they may have been selected for hunting, these explorations, and then in the end, so now they are just like our household pets. And most of the studies in dog behavior, they are very human directed, right? So for instance, in this first panel, there's this paper that looks at these yawning of the dogs, right? I mean, how much these social cues drive yawning, and whether yawning has a transforming capabilities. If would the dogs yawn if it sees a human yawning, and so questions like that. The other major question that people study in the second panel here is pointing behavior. So you will have an experimenter who's wearing these blinds because he doesn't want to detract the dog, and this lady here, she would point at one cup or the other cup and basically see if the dog can follow her pointing gestures, right? So it's how obedient your dogs are. And a lot of studies in dogs, they would ask humans to score in a scale of 1 to 5, for instance. Is your dog interested? Is your dog anxious? Or does your dog need space? So these are all very human emotions, right? And the idea is, to what extent we all know that we love to talk to our dogs, but to what extent of that is innate, or what extent of that is the general repertoire of dog behavior, that's questionable. So when you look at a dog in a field, so this is a pointing dog, and so these are hunting dogs selected to do trials. The idea being the dog runs and it focuses on a particular spot when it finds a bird, right? So, and I'll show you the pattern. So the dog runs. It's very active. It's highly energetic. It runs, it ignores the guy. And there are these complex movement patterns where when it's running, it suddenly turns, right? And it catches a whiff, it turns, it gallops. And then when it gets closer to this bird, which is in the field, it stops, and then it starts walking. And when it's really close to the bird, it does this very characteristic pointing behavior. And I'm sure in India, growing up, I saw it many, many times. Some dogs would do this naturally. Like they would take one of their paws up and look at a certain direction. So this is a very elaborate movement pattern. And when I was doing my grad studies, I wanted to look at this more carefully in a slightly more manageable setup. So this was my experimental field where, so it's quite a big field. So I gave them enough space. So this is 210 meters by 210 meters. And I had this designated path that the handler of the dog, or in those cases, some of the owners of the dogs would walk without issuing any commands. And the dogs would just run around. So dogs were free to run around for as long as they want. And what I did, I put tiny GPS collars on the dogs' collars so that I can track their movement. And so this is the tracking of those dogs. And if you see, I ran a lot of dogs, but sometimes it felt like some of these dogs have running patterns that are slightly more different than the other, right? So I mean, this is, I think, about 10 dogs running in the same field, superimposed. So a spaniel runs slightly different than maybe a pointer. And a house pet basically doesn't care about running. It just runs around the handler while the handler is walking. So this made me think that I might be able to look at this a bit more carefully and extract a bunch of behaviors from this. And the way I did it is first plot the movement. So these closed circles are the movement of the handler, of the human. And these 1, 2, 3, 4, 5, 6, 7, 8. So imagine these are 8 seconds, because I was collecting data every second on the GPS coordinates of both the guy or girl and the dog. And so these open circles are the dogs. As you can see, the dog's run is a lot more circuitous. It goes this way or that way, and it turns around. And the different things you can measure at a specific time how wide the dog is from the handler, which is this A. You can measure, say, on this movement from 4 to 5, what is the angle of the dog? And you can extrapolate that by saying, well, if the movement of the human between 3 to 4 was straight and the movement of the dog between 3 to 4 was on this right angles, so what is the angle between? And then you can also look at how sinuous or how straight or unstraight the dog's run is by measuring these distances between points. And to put it in a more human terms, you can measure the speed of the animal, you can measure the distance of the dog from the animal, and you're measuring it at every single data point. You can mention the fractal dimension. It basically is how loopy the dog's tracks are. So quartering ability is something that I picked up from the field, which basically means, is the dog paying attention to the handler while it is running? Basically, you want a high quartering ability where the dog runs in front of the handler. So it knows that the human is behind me rather than going all around. And then some angular variables, which is like the dog wants to run forward and how consistent it is at running forward. And so when you get all of those data, and so I'll put tiny panels here when kind of go back to this idea of machine learning. And so we are talking about this unsupervised learning where I'm not feeding in anything to the computer, and the computer is just looking at all of these data and just spitting out information. And you start with the raw data in any behavioral experiments. You extract variables And although here this is variable extraction from open field, which is my experiment, the idea is same for most of the behavioral experiments these days. When you have a lot of data, you want to see, are there relations in those data? You want to reduce the number of variables that you work with, and then you kind of reassemble all of those variables to see, are there hidden things that are going on? And the reason I say it's signs of correlated response of behavior in domestication, this idea was, as the dogs were being domesticated, certain features started to develop, right? And you would have heard of droopy ears or the coat color changing to black. And so those are morphological correlated responses that happened with domestication. But there was this idea that no correlated responses occurred in behavior, and people are still doing research on that. But most of the behavior they're looking for or they're looking at are not, I would say, represent the innate nature of the dog's behaviors. And so, at least in my very limited experiment here, where I just tracked dogs and extracted variables, it does look like some very distant variables are fairly well correlated. For instance, if you look at here the mean speed, and this is a lot of data, you don't have to look at all of this, but if you look at mean speed, the first one, and then path overlap, which is the last one, right here, it's very strongly negatively correlated, right? I mean, and you can understand that. I mean, it makes sense that these variables are correlated. I mean, if you're running very hard, then maybe you have less of a path overlap. Or if you are quartering, which is the human oriented features, then you have a little more mean speed. So there are some features in behavior, if you focus on the behavior, they can be correlated. So that was the basic idea, that and people, of course, you can extrapolate that some of the behaviors, when dogs were getting selected, or humans were selecting dogs, or they were selecting themselves, both the ideas are common these days, that these may be some of the features in the dog human co-evolution, I would say, may have featured higher than some other variables, because they were initially domesticated to carry out a bunch of works. And so don't focus on all of these numbers, but what you should focus on here is in this red box, which is person variance explained. And that just means how much variance from your data can be explained by reducing the total number of variables. So I started with 18 variables. And you see here, by using these only 4 variables, I can explain about 81% of all the variance in the data, right? And so these are called principal components, which basically takes all of these variables and kind of mishmashes them and gives you an underlying latent variables. And the cool thing you can do with that, since each of these axes are made up of multiple variables, you can be imaginative and you can give them names, right? So for instance, here I call it PC1, I call it energize exploring, because here in the bowl, all the speed and some fractal dimensions are related to this variable. And for instance, third one, I call it handler focus, because the high variables in these ones are relative variables, which are human directed. So you can be more imaginative and call these latent variables, but the idea being you can take all of multiple variables and reduce, because imagine the more you reduce the number of variables you work with, the less analysis that you need to do, and that helps you bring out more latent features, more hidden features in the data, right? And these 4 variables, when you just take those, and this is again unsupervised learning, I'm not telling it what to make of it, I'm just feeding this information in the data in the computer. And so these circles are, so you see 3 different colors of circles here: red, blue, and green. So those correspond to 3 different types of dogs. House, one type of field dog, the other type of field dog. And based on those 4 variables, you can see they cluster. And clustering is just means you take these data, and without knowing who belongs where, you put things together in a meaningful way. And even though in the clustering, so here you have hunting or field dogs, and here you have house pets which cluster out. You have some cluster that have both field and house dogs, which makes sense. Because there are no specific separations, even though breeds are a um, human induced feature, there are some latent behaviors that are still persistent between those breeds. And what you see here in the inset is, if you look at spatial axes and social axes, which is basically first and second, there is a lot of overlap in behavior. So when people talk about breeds and dogs, they say, okay, this breed is more aggressive and the other one is more work friendly, or this breed barks more and that breed doesn't. So there are a lot of overlap. So you have to be careful when you are saying, okay, one is better than the other, or they are different. And I think that, especially now in the current situation, this is more important than ever, that there is a lot of overlap in behavior, in morphology. And even though things look very different from the outside, if you really look closely, things are not that different. Things do overlap. And so I think the take-home message from the part 1 is, you can use computational analysis, but you also have to look at a lot of traits. You have to do multivariate analysis to understand some sorts of behavior. The signatures of domestication happen in behavior as well, if you're quantifying them. And genetic association studies. And because, let me go back to one slide, people always do these high throughput genomic studies, genetic association studies based on these breeds. The idea is, I take 10 breeds and I sequence their genome, and then I say which genes are associated with which breed, and they extrapolate that's is also associated to behaviors, which I think is a slippery slope. And one of the reasons why the genetic association studies of behavior has really not gotten much success in the last 20 years or so. And so genetic association studies of behavior, you need to really be mindful of overlaps of phenotypes. You need to quantify behavior really carefully in order to really get into the genetics of it. And that is difficult, given if you're working with something like a dog, which is very, very difficult animal to work with, because you have IACUC, you cannot dissect a dog's brain, you cannot do mutational studies, you cannot do functional genomic studies. It becomes really, really difficult to do all of these in dogs. So while I was finishing my studies with dogs, I was thinking about these things. So how to really understand behavior in a more manageable system. And that's when I joined Ed's lab, who was working in Drosophila and fighting. And I will show you this pattern again, which was lunging, which you saw before from the top. And these are male flies, because you see like how the butt is like black. And so these are the male features, male phenotypes. Females have ovipositors. And this is a slowed down video. So what you would see is this guy goes up in its hind legs and snaps down on the other male fly. And I'll show you again. This is like slowed down, I think 20 times, and it's a very fast behavioral pattern. And those lunges dictate most of social interactions that come afterwards. And when I was studying in Ed's lab, I found out that development really plays a big role in performing these features in behaviors. So what you have here, so bully just means it's a selected line which are hyper aggressive, and Canton S the way they name it, it's the flies are collected from a place called Canton in Ohio, and so they are just called Canton S. They are the S strain of Canton flies. So these are wild type flies, quote unquote wild type, because they have been lab reared for many, many generations. So when you select for hyperaggression, which are bullies, and if you focus on these total lunges, you would see that at 25 degrees, when they're reared at 25 degrees, which is room temperature here, they do a lot more lunges than wild type flies that are reared at 25 degrees. But if you rear the flies at 19 degrees, it seems like even though these bully flies, these are isogenic flies, by the way. And by isogenic, I mean, for the most part, all of their genes are the same in these strains because they have been selected and they have been isogenized, even though they are isogenic. So they are all same genetic material between 25 and 19. But if you just give them different developmental temperatures, their behavior changes quite a bit. And this happens specifically in the bully line, and it doesn't really happen that much in the wild type strain for some reason. So there are some mutations that were selected that are temperature sensitive, and that gives you a modulation feature in the behavior. And so I use this information to do what a lot of people were doing that time, which is called the RNA sequencing, which is a big data science. And I mean, I call it big data, but it's really not that. Everybody's doing it these days. It's not that difficult. And I looked at which genes are differentially expressed. And I found at the intersection, if you use this developmental feature, you only had about 9 genes that were different. And when you quantified those genes with qPCR, you had only 6 that were significantly different and matched your RNA sequencing data, and only 1 of them matched between different genotypes. And I showed that if you just do a correlation, relative expressions of those genes can be correlated to how much the animals are fighting or how quickly they start fighting. But with this information then, and this is also a very common feature in behavioral genetics. So just to give you a very quick view, so genes are expressed differently in different cell types. So a neuron would express neurotransmitters, a liver cell will express something like alcohol dehydrogenase. And in gene expression, you have, and all of you have very strong molecular biology background, so I won't belabor the point. You have this mRNA that comes out, which becomes a protein. But the small interfering RNAs, which is the RNAi approach, they form these clusters, these complexes, and then that drops on the target mRNA, and that reduces the expression of those proteins. So this is a very classic gene knockout experiments. And what I did in my experiments, so I downregulated 1 gene only in neurons, and then I found that changed social behavior, right? I mean, so this red square is my experimental, and these are my 2 parental controls. In all of these cases, I saw differences just by reducing 1 gene only in neurons, expression of 1 gene in neurons. And then it also changes the outcome of fighting, because the animals where I reduce this gene only in the neurons tend to be more aggressive. And of course, they tend to win more percentage of their fights. I don't know how much time I have. I'm almost getting done. So, and this is not uncommon. And so one of the reviews that we did, developmental windows do play a big role in modulating behavior. For instance, here, if you have a specific portion of the brain have low density of certain types of neurotransmitters at specific windows, that changes the total amount of sleep in adult behavior. So developmental windows like Florence, who focused on developmental windows and how that affects future behavior, you can see that there are now these molecular studies that are looking more and more into it. And so the take home is developmental windows are important. So not just nature, but the experience is also important. Big data science need careful strategies to focus on manageable number of genes, and selective expression of genes in neurons can change behavior, right? And so I'm just going to quickly go through this. So this is the first clip that I showed you, where two flies fighting. And what I want to focus on is maybe, let's go here, maybe do tracking, right? So I showed you in the beginning the two mice being tracked. So here I'm tracking these two flies, and although this is a very complex set of behavior, you can see it maintains the directionality of the fly, the angle of the fly, and once you have that, you can predict these lunge behaviors. So in the bottom here, you have these red bars that are, so this is all the machines predicting, but this is a portion of supervised learning. I have taught the machine how to predict behavior. And every time there is a lunge, you would see with the corresponding red line, the machine predicts fairly well when the lunge is happening. And this is my automated pipeline, and I think it's almost like an hour. So I'll quickly finish. So I basically take recorded video like I showed you before. I run it through. So this green one is my pipeline. I run it through a tracker. I do classification of lunges, and then I extract lunge numbers. And you need to be mindful of what the machine is telling you. So this manual, and so this is a part of supervised learning. So this manual is what is happening, and I count it manually by observing videos. And this classifier is what the machine telling you where each behavior is happening. And of course, sometimes there are misclassified information, but you need to make sure what percentage of those classifications are misclassified. And both of the cases, it seemed like the machine is doing fairly well. And you have a pretty strong correlation of what a human observer does and what the machine is spitting out. So it's working properly in the supervised learning. But what I really want to focus on is this, where aggression persists. Using those machine learning approaches, what I showed that behavior, generally aggression was studied for 20 minutes, maybe 30 minutes. But here I fought flies for 24 hours, right? And so I showed during the daytime, there is a lot of fighting in same pairs. And I could only do this for so many animals because I was using machine learning. If I had to manually sit down and count all of these for 24 hours, that would almost be impossible. And using the machine learning, I also showed that there are these rhythmic features in behavior. So during the day, the flies fight a lot more. So flies are crepuscular. So their activity of single flies is crepuscular. They're active early in the morning and then in the dusk. But aggression followed kind of like a reciprocal sort of a pattern, where at dusk they really reduced their aggression a lot, and that persisted both in light dark conditions and in complete darkness. So the idea was, if animals don't see each other, they don't fight. But clearly not, because I do also my videos in complete darkness, and the pattern seems to persist. And that persists it matches sleep more than activity, which was kind of surprising, that you would expect fighting behavior to proceed with matching activity levels. But for some reason, I see the sleep levels kind of match my aggression patterns. Which is also not entirely surprising, because so these are all the neurons that are mapped, and people have mapped fly sleep neurons for a long time. So there are wake promoting neurons, which makes the fly wake up. There are sleep promoting neurons, which make the fly fall asleep. and some of them are uncharacterized, but they make wide connections to different parts of the body, different parts of the brain, and some of these connections, of course, are dopaminergic and different other amines, which also are important in aggression. So the take-home message is, so think about machine learning as just a simple tool like PCR, right? I mean, everybody does PCR. Uh, in zoology departments, you would have to do some PCR. You have to do, for instance, centrifuges. But more than the method itself, you have to focus on what questions are being answered by those methods, right? I mean, machine learning is becoming the new favorite tool, but it is really what question you can answer. I mean, if you can't write a paper on PCR anymore. Like there cannot be a paper said, oh, I figured out different methodologies of doing PCR, which doesn't really is not that interesting. But what you do with the PCR or centrifuges or microscopy, they can be really interesting. So behavior persists long after one stops looking, because you can leave animals and stop looking. It doesn't mean it stops them from behaving. It really goes on. And behavior is guided by multiple modalities, and which means it's not just vision that guides behavior. There are odors, there are chemoreceptors. All of these can modulate behavior. And so this is let's see what time it is. And so this is just like the third part of that machine learning thing, where I give it information in real time. So the idea is, if a fly here in these tubes, if a fly falls asleep, the machine here records it in real time, sends a feedback loop, and turns these cables to wake the animal up, right? And so this is all happening in real time. And you would see here, this guy, actually, I think it's this guy. It's standing, it's not moving, and then the rubber turns. If it's standing, this rubber will turn, and the animal will wake up. And so that's what I mostly do these days. And this is what my rigs look like. You have multiple machines, and here for many days, so this green is my experimental. For many days, I only wake the animals up just when it starts to fall asleep, right? And so here are my 2 cents at the end of this kind of long talk. There is a revolution that's happening in animal behavior. How people are quantifying animal behavior and looking at it, that's changing. You need to focus on multiple behaviors to really understand complexity. There is big data in behavior, but you have to look at those big data and you have to make meaningful observations. And conventional ideas, just those stories, do need constant research. And I think that's it. And if you have any questions, I'm happy to take it. I don't know. I think I'm a little over time, but
Hello.
yeah.
Hello. Yeah. You are really on time. Yeah. Okay, Buddha, thanks a lot for very much knowledgeable and excellent session. We are really thankful to you. Okay, and now the session is open for questionnaires. I find if I have some few questions. Okay. One question came that is it possible to predict human behavior through machine learning?
That's
Is it possible to predict human behavior through machine learning?
That's a good question. And there are different levels of that answer. I would say, yeah, can you hear me? Yeah. So I would say people are already trying to predict human behavior. And you see this already. Everybody uses Facebook, right? Everybody sees ads on these Facebook. So a very common these days feature of machine learning in predicting behavior is in behavioral economics. So things you search for in Google, for instance, you start seeing their ads on your social media, like Facebook. And there is not a single person sitting behind and feeding you these apps, And as you're searching, even now, as you're talking on the phone, those snippets of information are being coded somewhere. So nobody lives a private life anymore. Everything, every data is out there. And now they're using it for behavioral economics by feeding you things that you might want to buy. Uh, but more less market level approaches of human behavior, predictions of human behavior is slightly trickier, because humans are a little unpredictable. But there are a lot of studies that are going on right now in doing predictions. I mean, like I said, behavior or feeding them machine learned information is already there. It's behavioral economics. You already see more ads. You Googled, I don't know, if you Googled soccer jerseys, you will get ads of soccer balls, for instance, no? Because they would learn that guys who are searching for soccer jerseys are more likely to buy soccer balls if you give them 5 ads of it. Anyway, so that's kind of what we are doing these days.
Okay, okay, thank you. Thanks for that. Another question from Piali Chakraborty, I can see. How we should choose animal for behavioral study if we are doing PhD? That means you want PhD. How we should choose animal for behavioral
Yeah,
study?
uh, I would say, uh, if you're doing a PhD, you should study the animal that your PI is studying. Uh, I did not do that. I studied something very different
Yes,
from
yes.
what my PI was doing, but I would not do that again if I were you. I think while you're doing your PhD, you have limited time. You should learn what your PI has learned over many,
Hello.
many. Can you hear me? Hello? Can you hear me, Atre?
Yeah, yeah, yeah. The sound
Yeah.
is cracking sometimes. I think
Yeah, it's
the
breaking up
connection.
sometimes.
Yeah, yeah. Now it is better.
Yeah.
Sound is
So I would,
connection error from my front. Connection error is
yeah.
happening. Hello,
Yeah, yeah.
hello.
I hear you. Can you hear me? Hello?
Yeah,
Yeah.
yeah, yeah,
So
yeah.
no,
Yeah. Now it is
I would
audible.
say,
Yeah, please.
a people would tell you to study the animal that you like the most. But I would say you should study the animal that your PI is studying. But I would tell you also this: while you're doing that, have your own questions that you can then pursue. So while you're studying an animal,
Yes, yes, yes.
you can do it in a more ecological way, which I think there are very strong labs in India that are doing that. You can do behavioral, very strong labs also that are starting in India. Try to learn as much as you can from those labs, because those are really good, but also have something that you are interested in that you can then pursue while you're doing your postdoc work, and where you're a little more independent. Flies are great to do behavior because so much of it is known. If you're interested in the genetics of behavior, the neuroscience of behavior, but if you're interested in the complexity of behavior, you can work with something more exotic, I guess. Uh, but personally, I'm a more question-driven person, so I am interested in specific questions, and that's what I would suggest. Learn what your PI knows in your PhD. That would be my two
Hello,
cents.
hello, hello.
Yeah. Atri, can you hear me? I think you are breaking up a little bit on your end.
Yeah, yeah. Actually, yeah, yeah. Because it's cloudy now, and because of the weather,
Yeah.
the sound is cracking. Connection error is happening. Uh, Okay,
Okay.
okay. I can see a few questions now. Uh, okay, another question from one of my sir is he is asking. He's telling that your genetic analysis is interesting, but do the genes are universally implicated in behavior? in behavior of animals or even in human, or just its fly behavior?
Oh yeah, that's a very good question. So this
Will I
gene,
repeat?
is a neurotransmitter transporter. So although, and this is a family from, it's a large family. It's called SLCs, so solid carrier proteins, which do neurotransmit transporters. Humans don't have the exact same gene, of course, as the one in flies, but they do have homologs on other transporters, and transporters are important in carrying out a lot of social behavior. So this is, I think, my gene was SLC38, and there are 40 different types of just solid carrier transporters. So um, some of these transporters are now being studied in neurodevelopmental disorders, which do have social behavior implications, but not directly. The gene that I studied has a human homologue. No.
Hello, hello,
Yeah. Actually,
hello.
yeah.
Connection is creating huge problem. I'm sorry. I'm sorry. Okay, hello. Okay.
Yeah. Can you hear me? I can hear you.
I think it is audible to the viewers. Yeah,
Hmm.
I hope that it is audible to the viewers. Okay, okay. The next question. The next question. What is the costing of an behavior animal behavior tracking set
Oh, yeah. So these
of
days it is.
animal behavior tracking set?
Yeah. So I
You
would
can
say
tell.
these days it's not that expensive anymore. Of course, you can go big and have a lot of money and spend a lot of money, but even in India, uh, you have these Raspberry Pis, which here now I think are $20. So that's like maybe 800, 1,000 rupees in India. A Raspberry Pi. I don't know what the exact number is. You have these tiny Raspberry Pi cameras, which are again here they are like $16. So that's maybe 700, 800 bucks. So you can spend $2,000, maybe 3,000 rupees, Indian rupees, and maybe have a rig to track. So it is becoming very affordable. Of course, you won't be able to do the really
Okay.
high 3D, high contrast imaging, but if you really want to do tracking, there are many ways to do it very cheaply in India. And also these GPS record transponders, trackers, they're also very cheap now. I don't know exactly what they are, but you have these wearable devices, right? So I mean, you have these Fitbits, you have these all of those GPS trackers that people wear for health tracking. It would be in those ballpark. So it's not, if you want to do tracking experiments, it's not going to break your bank. You can do that for fairly cheap, I think. Atri, Atri is gone. Atri is gone.
Hello, hello. Am I
Yes.
visible now and audible also? Am I hello?
Yeah, it's much better.
Ah, oof. It's creating huge problem.
That's okay.
Okay, lots of it's really a excellent session. Is there any Autindriya Shane asks, is there any possibility in future that one can easily control the behavior of other by machine? Can we eliminate anger, depression, etc., emotional state by knockdown particular gene?
Yeah, so that's a very good question again. Um, to knocking down and knocking up particular genes in humans, I don't think we are there, or we will be there in a very long time, which is social behavior. With social behavior, anger, depression, and all of that stuff. When it comes to modulating states with machines, we are already doing that because you have these, for instance, you have these Parkinson's patients now who have this really Parkinson's chorea. We have this sheikh who have difficulty in carrying out daily daily regular work, but now you have these deep vein or deep brain stimulus, right? So DB technology, where they basically put an electrode in deeper in your brain, which is a machine, and when you turn that on, movement patterns become very easy, and that have been going on for many years now. So that's just changing these motor patterns in humans. When it comes to regulating or down regulating genes in humans, that's more difficult. Some of them, there are some technologies people are using these days um, to do some gene specific therapies, but that's in disease, like in cancer, very advanced cancer. uh, I think there are some of those experiments, some of those studies that are going on in HIV as well, gene knockdown studies. But I don't think, both experimentally and ethically, there will be uh, gene up or down regulations in humans in the near future or ever. There will be a lot of problems if you are trying to control mood uh, using gene switches, for instance. I mean, people already do mood, um, I would say, quote unquote, control using these antidepressants, which are not that great. They have a lot of side effects. But single genes, I don't think we are there. I don't think we should be there, actually, if you ask me. Okay.
Okay, thanks. Thank you. The last question. Does a pet can affect its owner's behavior?
Good question. I would say yes. It definitely makes the owner more responsible, I would think. Like if you forget to feed your
Okay.
dog and take it out on a walk, that dog will be very
Yes,
unhappy.
yes.
So, in some sense, yes, and I
Yeah,
would say
very
that
true.
is the behavior. So yeah, I would, and in other levels too. I think people are studying these social dog human connections now in these very cognitive labs, and they're showing there are changes in human patterns if you have animal companions, right? I mean, there are now these companion dogs which are given to uh, uh, humans with like neurodevelopmental conditions and non-neurotypical disorders. So they do help human behavior. Having companion animals, it's very important. Okay.
Okay. Okay, thanks a lot, Buddha. Thank you. And we hope that in future we also get you whenever we call you.
Yeah, yeah. Atri has my email. So if you
Okay.
have any more questions, you can just email Atri and then
Yes,
she can
yes,
forward.
yes, yes.
Yes, absolutely.
Yeah, that's a good way to connect you. Yeah, yeah, yeah.
Yeah,
Obviously,
I'm happy to
that's a very
answer.
good way
Yeah,
to
yeah,
connect you. Yeah.
yeah.
Hello, 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 hello. Yeah. Okay, now we are at the end of the program. It's a valedictory session, and a vote of thanks will be given by my colleague Shatarupa Pal. Shatarupa, please, it's over to you. Hello, hello, Shatarupa, can you hear me?
Hello, Achidi, I can
Hello,
hear you.
hello,
Hello,
Shatarupa.
Achidi. Hello, Achidi, I can hear you. Hello,
Hello, hello,
hello.
hello.
Yeah, I can
We have a
hear.
huge problem. Yeah, yeah, yeah,
Yes, I can
yeah,
hear you,
yeah.
Achidi.
I can hear you. Okay, okay, okay. Please proceed. Please proceed. Okay.
Thank you, Achidi. First of all, a very good afternoon to all of you. Honorable dignitaries, respected guests, and dear students and participants. On behalf of Michael Mudusudhan Memorial College, its entire fraternity and organizing committee, I feel immense pleasure and want to express deep sense of elation for having this opportunity to express the vote of thanks to all of you. Firstly, I wish to express my sincere gratitude to our Honorable Principal Sir Dr. G.M. Helaluddin for entrusting us with such a huge responsibility to organize this international webinar, and of course, for guiding and encouraging us through this. I want to convey our deep regards to our eminent resource person, Dr. Buddhaditya Chaudhuri. Thank you very much, sir, for sparing time from your busy schedule and for blazing our webinar by dispensing your valuable knowledge. Thank you very much. Through your examples, we are able to recognize us and allay your apprehensions regarding the applications of machine learning in zoological field. Very much, thank you very much, sir. I take the opportunity especially to express my deep regards to our respected and beloved head of the department, Dr. Atri Shahana, for always being a resolute support for us and for having confidence in us. Thank you very much, Atridi. My deep sense of appreciation to all of our participants who chose to be live with us and attend the webinar with great enthusiasm and have made it a successful event. I thank Mr. Swadhin Kumar Mandal, Assistant Professor, Department of Sanskrit, Michael Madhusudan Memorial College, for the technical support and for helping us to webcast this webinar through the YouTube channel. Last but surely not the least, our departmental students for being so enthusiastic. Once again, I thank you all of you for being with us this afternoon. Have a wonderful time ahead. Over to you, Achidi.
Thanks a lot, Shadrupa. You took the responsibility of giving this vote of thanks. Okay. Now it's time to say formal goodbye to all of you. Thanks a lot for your kind support and patience. Hope we will get you afterwards if we arrange these types of webinars. Okay. Thanks. Thank you very much. Thanks, Buddha.
Yeah. Thanks so much. Yeah. Thanks a lot. Okay.
Thank you, sir.
Okay.